I will see that on paper also. It's good.
Yeah, have a look at On's paper. The formulation we made were for oral delivery, but
Okay, sorry for the interruption. Can we start the session? Okay. So a very good morning to all. I, R.K. Singh, welcome you all on day 4th of IBRO-APRC Associate School on Advances in Nanoneurotherapeutics and Neurological Disorders, organized by ISFCP College of Pharmacy, Moga, and sponsored by IBRO-APRC. Today we have with us our speaker of the day, Dr. Tushar Kumeria from Australia. He is a senior scientist lecturer at the School of Material Sciences and Engineering, University of New South Wales, Sydney, Australia. He received his PhD from University of Adelaide with a doctoral thesis in Medals and Dean's Commendation Letters in 2015. He spent his 2 years in University of California, San Diego as a postdoc fellow in Professor Saylor's lab, and after returning to Australia in 2017, he received prestigious early career fellowship at University of Queensland. He has co-authored over 98 peer-reviewed publications in top-tier journals in the field of nanomaterials, biomaterials, drug delivery, and consequently have attracted more than 3,760 citations on Scholars with an H index of 39 and more than 3,000 citations in Scopus with H index of 35. He has successfully secured over $4.1 million as a research grant, including NHMRC Early Career Fellowship, ARC Discovery Project, Rama Kiyoti Health Investment Grant from US Department of Defense grant. He has also served as a Vice President of Australian chapter of Controlled Release Society. He has also received over 15 awards, prizes, and recognitions from reputed organizations. So we all welcome you, Dr. Tushar Kumeria.
Thank you, Arti. That was a good introduction. I can start sharing my screen now. Let me know if you can see it. Here. Can you see my screen now?
Yes, we can see.
Yeah.
Okay. Right, this is a new platform for me, so it's taking some time to figure out. All right. I hope it is in presenter mode now.
Yeah.
Right. Okay. So we can begin now. Thank you, Arti, and thank you, Charan, for inviting me. I've known Charan and Arti since 2018 and 2017, 2018, and it's been a pleasure. And we've collaborated on a few things, and it was a lot of fun time when Charan was here in Australia. I hope he can come back. We can collaborate on things if you come back, Charan. So my research involves we make a range of different type of porous materials, and we use those porous materials for many different applications. We use porous materials for biomedical applications. We use porous materials for environmental application. Today, I'm only going to talk about 3 major applications of a few different type of porous materials we make. So I'll talk to you about drug delivery application of porous silicon dioxide based materials, their tissue imaging applications, and tissue engineering applications. So let's dive right into it. Remember this photograph on the first screen. You see these colorful images. There's a reason that they are here, right? So remember that I showed you something in the first slide. So, right. So everything we do in the lab has a few guidelines that our systems follow. And why do you see this picture of the cover of iRobot, the book from Isaac Asimov? This is the same book on which the iRobot movie is based on, right? So the reason it's here is because this is the book in which Isaac Asimov in 1950s gave us three main laws of robotic, right? So the first law of robotic was that the robot must not harm or injure a human being through its action or inaction, right? Second is it must obey a human being unless this conflicts with law number one. And third is the robot must protect its existence unless it conflicts with the other two laws. Similarly, we need some, rules and guidelines for nanomedicine. You see so many publications and so many, research happening in nanomedicine, and only a few handful of the products that have come out. On day one, Professor R.K.Gupta, he mentioned something very, very important. Do not work on nanomedicine if you don't have a purpose. Working on nanomedicine is not the purpose. The purpose is to solve a particular problem, right? If the nanomedicine can solve a particular problem, use the nano platform. Otherwise, there's no need for it. You can work with other type of platforms. So in my lab, we design systems which are useful and adhere to certain principles to make sure that the nanomedicine platforms we make, they are safe, efficacious, and targetable, right? So three principles that the nano carriers in my lab that we make follow is they are very good at their function. So if the particles are made for delivering a therapeutic payload, they deliver the therapeutic payload efficiently. If they're targeted for bioimaging application, they do that bioimaging application. Or if they are for a combination, then they provide theranostics, gain very efficiently. Second is we can modify the surface of our particles to decorate them with targeting moieties, right? So that's the second, principle or law that our particles follow. And the third is that any type of particles you make in the lab, they must clear out from the body efficiently and safely without leaving any harmful trace, right? This is why lipid nanoparticles based vaccines were the only one and the first one to be approved, because our body knows how to safely and efficiently clear out those lipids. Nothing else, is in the market for, or could come to the market so quickly, because testing and long-term testing is not done. It takes ages to know what would be the long-term safety impact of other type of particles. So this is where, you see a lot of paper publications on carbon nanotubes, graphene-based systems. They might be okay for, I might be upsetting a few people who work on those. They might be okay for applications where you require specific, to use some specific properties of those materials. But they are not good as a carrier. Why? Because they don't degrade in your body, right? If you take silica nanoparticles, they are listed as GRAS compounds or GRAS materials by FDA only for oral delivery, and nothing else is approved. They are not approved as injectable formulation. There's a reason for it. The reason is we don't know what's the long-term impact going to be. And we don't want to end up with something similar to asbestos. Asbestos as chemical material is magnesium silicate. There's magnesium and silicon dioxide, right? And the reason it just, it's so toxic and it's so harmful is it does not degrade in your body. It can stay, if you inhale it, it can stay in your lungs for 40, 50, 60 years. And that's why that long-term safety is important. This is where our materials, they tend to follow these three principles. And the third one is the most important, that the particles clear out from the body into something safe and non-toxic, right? So in my lab, we make a few different type of porous materials, and we use them for a range of different applications, as I mentioned earlier. So for drug delivery, tissue engineering, and most of the biomedical application, we use porous silicon. Let me pick up a pointer. So we use porous silicon. We use porous silica a lot for oral delivery application. We've recently made the silicon micro rod for tissue engineering and skin delivery application. We work on composite nanofibers for tissue engineering applications as well. And we make titanium nanotubes and porous alumina for separate application. And those applications involve tissue engineering, where these are used for, can you hear me? The screen went white. Okay. So, titanium nanotubes are mainly for the implant application. So we grow these tubes on the surface of titanium implants for localized delivery of therapeutics. Porous alumina mainly used for sensing application. We use porous silicon as well for sensing, but I have not put any example of it in my slides. Right. So today I'll mainly talk about these three: porous silicon, mesoporous silica, and composite nanofibers. So the key thing that if you remember in the first slide, you remember they were colorful chips that were floating around. The reason they were like chips is because the way we make our porous material. So the way we make our porous material is through a process called electrochemical anodization. And in that process, the substrate material that you want to make pores on acts as an anode. So in case when we want to make porous silicon, we start with a silicon wafer. And that is what you saw in the first slide. If we want to make pores on aluminum, we start with an aluminum foil. If we want to make pores on titanium, our starting material is titanium foil, or dental implants or orthopedic titanium implant. And the process is relatively simple. You have your working electrode, which is connected to a positive terminal. You have a negative electrode, counter electrode connected to a negative terminal of the power supply, and you have a suitable electrolyte. For porous silicon, the electrolyte that we use is hydrofluoric acid based. So if somebody wants to try it, make sure you have the proper safety equipment. This is one of the nastiest chemical that you can work with. What this process ends up doing is it selectively etches these nano-sized holes in silicon surface, and these nano-sized holy layer we can lift off and make particles for a range of different usages. So I'll show you a video of the whole process. So this is how we start. We start with the silicon wafer, we pack it in an etch cell, and we apply current in presence of an electrolyte, hydrofluoric acid-based electrolyte, right? So what we get is a porous layer on top. So this is a whole 4-inch wafer etched to form porous layer on top. So this is, one of the things that you need to make sure when you're making particles is how easy is it to make them, and can you commercialize the whole thing? Can you make them in large quantities? And this is something that gives us confidence, because these silicon-based fabrication processes are very, very well established. So every phone, every electronic device in the world uses this process. So processes are all very well set. I've shown you this 4-inch wafer. There are 12-inch wafers that you can etch and make particles in really large quantities at much lower cost. So the porous layer that is formed, you can detach it from the underlying silicon substrate. And the freestanding porous layers that you get, you can make it into smaller particles. So this is some of the particles that we form after, these brown looking gunk is what we get after breaking these into small pieces. And these are, since this was a flat sheet that was lifted off, you get sheet-like particles. And you can control the size of the particles by sonication time or milling time, or the way you etch. And the pore diameter as well, you can change by controlling the current density of the electrolyte, right? So these are the key features why we use porous silicon-based materials. Okay, so the main reason is we can tune the pores. We can get pores which are 2 nanometers, or we can get pores which are 100. Why do we do that? Because we work with different type of payloads. We work with payloads which are small molecule drugs. We work with payloads which are macromolecular payloads, such as proteins, peptides. We are also working now with nucleic acids, like RNA delivery, applications. So for those, we make pores which are much larger. We can tune the surface chemistry. We can have particles to show really highly hydrophobic surface. Why? Because then we can have particles which can efficiently load hydrophobic therapeutic payloads. And we can have surfaces which are highly hydrophilic to show surfaces which efficiently load protein-based stuff, or hydrophilic therapeutic payload. Depending on the application, we can control the surface chemistry. Other things are, we've tested their cytocompatibility, and many other people have tested their cytocompatibility across the globe. We know we can get really high loading capacity. Why? Because the porosity of these materials can be as high as 85%. So almost 80% of the structure is air. So you can fill that air pockets with your therapeutic payload. The other thing is we have really high surface area. When I say surface area is really high, I mean on the order of hundreds of meters square per gram. On an average, the particles we make have a surface area of 300 to 600 meters square per gram. Right? That's basically 30 to 50 grams of this material having equivalent surface area of a football field. So that is, a key advantage when you want to load drug or attach therapeutic payloads onto it. We can tune the release profile by either changing the structure of the pores, size of the pores, size of the particles, and chemistry on the pores. We know that these particles are biodegradable because we've done in vivo testing. There are other people, other groups around the world which have done in vivo testing. We've injected these particles into rabbit eye. We've injected these particles into mice, rats, and we've done their degradation studies, and we know that these particles clear out from the body safely, dissolving into something that your body uses as a micronutrient for silicon. So that material is silicic acid, right? The other key advantage of using porous silicon-based material made using this process is that they tend to be self-luminescent. So you don't need to tag a fluorophore onto it to visualize where your particles are. So if you're looking at in vivo, IV imaging or IV dosing of your particle or your formulation, you want to know where your particles end up. Most other particles, you would need to attach a fluorophore to know where your particles end up, whereas our particles, they are self-luminescent. So we don't need to tag them with any fluorophore. And what do we use these for in the lab? We use these for drug delivery. We do some tissue engineering in the lab. We do a lot of biosensing and bioimaging applications. And here's the first example where we've made a porous silicon-based thermosensitive hydrogel formulation for delivery of therapeutic payloads in the nasal cavity. The application that we are targeting here is for reducing the inflammation in the sinus, but we are also looking at nose to brain delivery using these formulations. So, we can have direct access into direct access to the brain via the nasal cavity. So why make this formulation? The simple reason is, if you take a hydrophobic drug and you make a hydrogel made from polaxamer or similar, polymers which form a thermosensitive network, your drug, hydrophobic drug, tends to crystallize out. Why? Because your gel is water-based. It does not like it. Whereas when the drug is loaded inside the pores of these porous materials, the drug stays as an amorphous form and can't form a long-range ordered, crystalline structure. So the drug stays as amorphous. So what that does is that allows, us to achieve much higher solubility of the drug, and it also allows us to have much larger loading of the drug inside the pores, right? So using this, principle and idea, we formulated porous silicon particles based polaxamer hydrogels, which remain as liquid at storage condition, 4 to 8 degrees Celsius. They are liquid. Once you increase the temperature to about 28 to 32 degrees Celsius, they become a gel. right? The second advantage of system like this is when you spray a nasal spray, which is solely liquid, when you spray it, a lot of it ends up at the back of your throat. So what this allows is this gels are forming a film inside your nasal cavity, so the drug stays in the nasal cavity for much longer time period. Right? So these were the main reasons that why we developed this formulation. The formulations we made, we wanted to have a gelation temperature of about 27 to 28 degrees Celsius. Why? Because mostly where this would be applied is either in hospital or in home settings, and in winter time or in peak summer times in Australia, the temperatures are set to about 22 to 24 degrees Celsius. So the temperature of your nasal cavity is overall lower too. So that's why we wanted to have a gelation temperature, which is not your actual nasal cavity temperature, a slightly lower. So we ensure that it gels out when you spray it in your nose. So we made formulation with 0.1 weight% equivalent drug, 0.2 and 0.5 weight% equivalent drug. To make these, we made first particles which had about 30 weight% loading capacity, 29 approximately. So we incorporated that 29% loaded particles to have 0.1, 0.2 and 0.5 weight% loading. What this comes out for this is about 13 milligrams of particles in 1 gram of gel. And given that almost 70 to 80% of these particles are air, that 13 milligrams is a lot of volume of particles, right? So what this tells us is even at 0.58% loading, we don't see much change in gelation temperature. And the drug we chose was mometasone furoate. There's already marketed formulation, hydrogel liquid formulation at 0.1 weight%. And if you ever buy it, you can see crystals forming at the bottom of the vials that you buy. So what we had, we tried two times the current available formulation and five times the currently available, equivalent drug amount formulations. And we don't see much impact on the gelation temperature. The second thing we did was we looked at how the drug releases from these formulations. So these are done in Francel. What you see is the formulations which have porosilicon-based mometasone-loaded porosilicon added to the hydrogel. You see much larger amount of drug being solubilized and released from the hydrogel, whereas when the drug is directly added to the hydrogel at the same equivalent amount, you see bare minimum amount of drug coming out from the system. And it's same for all three type of the gels. Right. So that gives us confidence. And the reason that happens is because the drug stays as amorphous as an amorphous molecule inside the pores. There is no long range order formation. I have not shown the data. We've done powder XRD on it to make sure that we see we don't see any crystalline drug in these when these are loaded into porosilicon particles. After that, we moved on to drug deposition into human nasal tissue. So we got human nasal explants from a hospital around us in Australia. We looked at how much drug gets deposited per gram of tissue. And what we found is when the drug is solely added directly into the hydrogel versus when porosilicon embedded drug is added into hydrogel, we see 19 times increase in the amount of drug that is deposited into tissue, which is very encouraging and very exciting. And the main reason is because the drug is more bioavailable and more soluble, right? We also looked at the toxicity of the drug or the whole formulation. So these are the histology slides of the different formulations shown here. And we also looked at LDH level. And what we see is only zinc sulfate, which is the positive control, which is known to harm the tissue, shows LDH release and damage to the olfactory epithelium lining and basal tissue in the human nasal explant. Whereas the negative controls and our samples did not cause any such toxicity. So we've already done this. We are now exploring nose to brain delivery potential of these formulation. So that's the that's the next step. And if you have any student who's interested in something like that, do ask them to reach out to us. I'm looking for PhD students if you have any. Let's move on to mesoporous silica-based particles, and I'll show you imaging application of mesoporous silica. So here I've shown an image of dendritic silica, but the particles we've used is MCM41 type. It's just, these look more beautiful than MCM41. MCM41 have just, straight running pores through in a hexagonal manner through the particle, whereas these have dendritic, porous arrangement. So what we do in our lab is, as I said, we make different type of porous materials. The first type of porous material I showed is made using a process called top-down approach. So we start with something large, we break it down into smaller particles. Here, the approach is the other way around. It's a bottom-up approach. We start with molecular size, organizing units to form a larger particle structure. So we start with the precursors, such as tetraethyl orthosilicate or methyl orthosilicate. We start with surfactants, which form the initial liposome or lipid, micelle, on which the silica condenses, hydrolyzes and condenses to form that particular type of structure. So in our lab, we make a range of different type of mesoporous silica particles. We use them for many different applications. So, I'm only showing you one. And even when we make mesoporous silica, especially for injectable formulation, we tend to convert silica into silicon. We tend to reduce silica into silicon through a thermal reduction process. The reason is we know that silicon dissolves out safely and quicker. Silica does too, but it's relatively slower. That depends on the thickness of your silica walls in the system, right? So these are some of the silica particles we've made. These are solid. These are MCM41 type. You can see straight running pores. You see these lines. Each of these lines is a pore. Each of these, dot is a hole or a pore running straight through the particle. These are mesoporous silica-based rod-like structures, and these are dendritic emissions. So we make a few, and I'll show you an application of MCM41. So in this case, we use MCM41 type particles and thermally reduce them to form porous silicon type particle or silicon reduced forms of silica. And we functionalize them with PEG and attached an antibody for targeted delivery of these particles into a tumor, right? First thing we wanted to check is, are we sufficiently able to reduce the particles or not? And do the reduced particle maintain their structure or morphology at all or not? So what we see is, yes, we can reduce the particles. They do maintain their porous structure, but they become much rough, which is expected. We were expecting something similar. We've converted other type of macro-sized particles, and we see roughness of the particles increase. So we expected this to happen with these particles as well. After functionalization, we don't see much change in the size of particles. Even after attaching antibody, the size of particle does not increase much. It's goes from about 60 to about 70, 72 nanometers. The application or imaging modality that we have used these particles for is called photoacoustic. So I am pretty sure everyone here is aware of what ultrasound is. In ultrasound, what you do is you shine ultrasound waves, you pick up the reflected ultrasound wave, on the transducer. Whereas in photoacoustic, as the name suggests, you use light to excite the ultrasound waves from your material or from your tissue itself. And to do that, you use a near-infrared pulse laser. And that pulse laser, what it does is it heats your particles momentarily. And that thermoacoustic vibration is within the ultrasound frequency range. And that is what we pick up. And that's why this process is called, or modality is called, thermoacoustic or photoacoustic imaging. So first thing we want to do is we want to look at if the particles by themselves show any photoacoustic signal after reduction. So our particles show a very strong photoacoustic signal at 40 decibel of gain at around 700 nanometer excitation wavelength. We've done silica particles as well. They don't show anything around here. Why? Because they don't absorb much in the NIR range. Silicon absorbs more, and that was one reason to convert it. Besides that, we know that silicon degrades out. The other thing is indocyanine green ICG is an FDA-approved dye, which is currently used for many imaging modality. It's not approved for photoacoustic. Photoacoustic imaging is not clinically available, but some hospitals do use it, through a local ethics approval. And that's the dye that is used. And the problem with this dye is that it degrades. This is not a quick process like ultrasound. It takes relatively longer, but gives you much higher definition, right? And gives you pinpoint information. And ICG is not suitable because within first 10 minutes or so, you see about 20% signal loss at the same concentration, whereas particles remain stable, much more stable in terms of their photoacoustic signal within that time period. So we were quite happy with these results. So we moved on and we did in vitro imaging. This is ICG by itself at same concentration, and these are porosilicon particles. And what we see is we see 6 times more photoacoustic signal from particles compared to ICG. We then moved into this is all in vitro. We wanted to see what happens if we inject these particles into mice. So we started with an ex vivo experiment. We had mice cadavers, so we subcutaneously injected particles. So PBS shows no photoacoustic signal, which was as expected. Silica particles, silicon dioxide, they don't absorb much in the near infrared range, so we don't see much signal. Whereas silicon particles, the reduced one, they show a very strong photoacoustic signal at the injection site. And one of my interests with these particles is brain imaging. Why? Because there are now companies like Fujifilm developing photoacoustic imaging systems which can look through the skull. They are in near IR window. Their frequencies are different, so they can actually look through the skull. In one of the ex vivo experiment, we took the mice brain out and we locally injected some of the particles in the mice brain. And what we see, you see these red dots is where the particles were injected in the mice brain. So these particles, show quite high photoacoustic gain, regardless of different type of tissue where it's injected in. Here it's skin, and here it's a brain tissue. After that, we wanted to see if the particles can be targeted or not, because we had an antibody that specifically targets a tumor. And we looked at, if the particles get uptaken by those specific ovarian cancer tumor cells. And what we see is we see much higher cellular uptake in the targeted particles versus non-targeted particle or particles attached with a SCRAM antibody. This is the in vivo data. What we see is this is 48 hours after injection into mice which had a subcutaneous tumor. And what we see is a higher signal from the particles which are targeted versus the non-targeted particles. And we also recorded the kinetics of the tumor uptake of these particles. And what we found was that the non-targeted particles, they accumulate in the tumor or reach the tumor much quicker, but they get cleared from the tumor as quickly as well. Whereas the targeted particles reach the tumor slower, but they get accumulated and stay in the tumor for longer. This is the main reason for this is the kind of antibody we have. The collaborator I work with, they are the one who developed these or discovered this antibody, and they know that this antibody takes some time for their own targeting imaging agent to accumulate. They use radio imaging labels. This was the first time somebody has tried their antibody with nanoparticle type imaging agent. And the difference between the particles which are targeted versus non-targeted is significant at 48 hours. So that's on bioimaging using silica particles. We are making other type of silica particles and their converts for tissue bioimaging. We are embedding different type of other modalities like SPR based particles into these silica particles, MRI imaging particles. and a few other type of different particles. That is what we are working on right now. So next thing I wanted to talk to you about is the composite nanofibers. Sorry, I'm going a little fast because I want to cover the all three different type of systems. But next thing I want to talk to you about is the composite nanofibers. And the way we make these fibers is very different from mostly reported nanofiber fabrication system. Most of the nanofiber fabrication system you will see in the reports is based on electrospinning. So what you need to do is you have a needle and a syringe pump that pumps the polymer solution, and you apply 10 to $20,000, 20,000 volts on the system to get these Taylor cone, stable Taylor cone and nanofiber extrusion. Whereas in our system, we just simply need a compressed gas. We need a rheology or viscosity optimized polymer solution, or solution of polymer with porous silicon in our case, and and simply need compressed gas. And the system is, it allows us to spray the nanofibers on non-conducting surfaces, whereas with electrospinning you can only spray fibers on conducting surfaces. So here what you see is, you see nanofibers on a paper, cover slip, a gloved hand, aluminum foil, just to show that the versatility of the technique. And we can get fibers which are aligned and non-aligned just by changing the direction of spraying. So with electrospinning, you need to have spinning a spindle which rotates to collect aligned fibers. Whereas in our case, you just simply need to change, if the fibers hit head on to the collector surface, you get random organization. If the fibers hit at an angle, a steep accumulation angle, you see fibers which are aligned in a particular direction. So that's another advantage, and this is what we've used for tissue engineering, especially engineering the nervous system, injury systems. So first thing we wanted to do is to look at the optimization of nanofiber fabrication. You see these dot-like structures, these are simply the polymers which, droplets which did not get converted into nanofibers. So we want to look at, can we get nanofibers more consistently and get rid of these droplets? So we wanted to look at what happens if the collector is directly placed under the nebulizer versus a bit off, about 15 to 20 degrees off. And we looked at different spraying times and different collection distances. So what's the distance between the collection plate versus the nebulizer tip? So what we see is offsetted, collection, and 25 to 20 centimeter away gives us the most consistent nanofiber fabrication with the lowest amount of these droplets. We still do see droplets, but I think it's much lower compared to when we have the sample kept directly underneath the nebulizer. We also looked at the nebulization time and if the fiber density changes, and as expected, yes, we do see a change in the nanofiber density. This is a customized program, MATLAB program we made. It simply tells us how many pixels are bright versus how many pixels are dark. So simply looking at fibers which are sprayed for a very small amount of time, you see a lot of bright pixel in the image, whereas fibers which have been sprayed for 5 minutes, the whole surface is covered. And this is an example of, these fibers. It's a fiber like mat, which can be used for tissue engineering application. And this is what these fibers look like in under a scanning electron microscopy image. So we also looked at mechanical properties of these fibers. Why? Because these are going to be implanted as scaffolds, and most scaffolds you want to match the mechanical properties of your underlying tissue. What we see is as you see in the previous image, you see an increase in the thickness of the scaffold with spraying time. That leads to a change in the ultimate fracture load or failure load of the nanofiber scaffold. The ultimate tensile strength does not change much, but the load at failure does. So that's that was something that was expected. Failure strain does not change much. It does go up, but it does not, increase. I'm getting some noise. Charan, can you mute? I think I'm getting some noise.
Can you mute? Dr.
Yes, Tushar.
Thanks, Charan. So we looked at elastic modulus as well. We don't see much changes in elastic modulus. Unexpectedly, it goes down for the fibers which are sprayed for longer, but there's no significant change in elastic modulus of these fibers. After that, we looked at how well do the cells grow on these fibers, and we grew Hackett cells on these and at 2 different cell densities. And what we see is cell attached very well on these surfaces. And, over time, the 2D tissue culture plate sees more cell growth and cell proliferation, or cell expansion, whereas our fibers, they don't see as much cell growth. But what we believe the reason for this is the seeding protocol. The way the cells are seeded on these fibers, we see some of the cells are not actually seeded on the fiber, but around the fiber in the tissue culture plate. So that's the reason our initial seeding number is lower, and that's why we see a lower number being on the fibers. As I said, we can make fibers onto any kind of surface. And one of my key interests is, can we make a scaffold directly onto a living tissue, nanofiber scaffold, which cannot be done with electrospinning because you need to apply really high voltages. So, but our technique does, and this is what this data shows. So we use compressed gas or ambient, compressed gas as a driving force. So we looked at what happens, if we spray these fibers directly onto the cells. So ambient air, when it's, so this is unmodified samples, so no treatment. This is when the cells are exposed to ambient air. This is when the fibers are simply blown with compressed gas and nothing else. This is when we spray dichloromethane. Dichloromethane is the solvent we've used for making these fibers. And this is when the liquid DCM is added. So you see when liquid DCM is added, almost all the cells are dead. Whereas when the DCM is sprayed using the nebulizer that we've customized, we see only about 2.1% of the cells dead. And when we spray nanofibers using the same nebulizer, we see only about 4 to 5% cell death, which is highly encouraging. And this is what we see in confocal live date staining images as well. So this is DCM itself, and these are our nanofibers directly sprayed onto living cells. We've submitted this paper to biofabrication. Let's see, fingers crossed that it will go through. But these are highly, very interesting and very encouraging results. We also, as I said, these are composite nanofibers. And most of what we do is, porous silicon-based composite. So these fibers contain porous silicon particles inside the, fibrillar strands. The reason to do this was so we can deliver therapeutic growth factors and therapeutic protein payloads. Why? Because incorporating protein-based growth factors into polymers, which are only soluble in dichloromethane or chloroform, is not possible. Your polymer protein denatures. So that's why loading a protein into these particles and then embedding them into the nanofibers adds an advantage that the protein is protected and it releases over time slowly. So this is a release curve, in vitro release curve of the protein-loaded particles. These are not from the nanofibers. This is just the particle. And what you see is over 200 hours, almost all of the protein that is released retains its activity when embedded in the nanofiber. So this is the release of lysozyme from nanofiber composite. And what you see is almost about 75% of the protein that is released from these composite nanofibers still retains its activity, whereas we've done controls where we have just the PCL nanofibers and we see nothing, like all the protein is denatured. We've looked at growing, the end application for these is nervous system growth or guiding nervous system cell guiding. So we've looked at growing cells and looking at if we can guide neurite growth along the direction of fibers or not. And what we see is we can get these are DRG cells, rat dorsal root ganglion cells. These are grown on a flat film, and what you see is these neurites growing in every direction. So there's neurite forming a corona around the cell, the central body. Whereas when you grow these cells onto nanofibers, which are directionally aligned, you see the growth of the neurites happening primarily in the direction of the nanofibers. So this is what you want in case of a nervous system injury. But this is a large cell. We also wanted to look at if much smaller cells would show the same behavior or not. These are Neuro 2A cells, which are grown on aligned fibers versus non-aligned or plane surfaces. And we see, a very well-defined directional alignment of these cells as well. So these are the kind of things we do in the lab for, brain targeting, for imaging of the brain, delivery of, therapeutics to the brain through nasal route. And we are also now working on IV delivery of cancer therapeutics. I have not put any data on it because we haven't done a lot. Probably in the next round when we have IBRO conference, I can show that data. We also do tissue engineering application targeted towards nervous system repair. So that is where I will stop, and would like to thank all the people and institutions and funding agencies which are involved in this or have partially or fully funded these projects. There's a lot of people to thank. I won't name all of them, but I should mention that Shrishti and Zach and Zanab did all the imaging work. Yvonne and Mike are my collaborator who have done all the nanofiber work and composite work, and Yahoo and Thomas and who else? John Hooper, they are involved in the cancer targeting and drug delivery work. So I should stop here, and thanks to Charan and Arti for inviting me to give this talk, and thank you for listening.
Thank you very much, sir, for a wonderful talk. Now the session is open for discussion. Yes, we have our eminent speaker, the next speaker of the day, Professor Pankaj Seth, and he wants to discuss something. Yes, sir.
Yeah, Tushar, wonderful talk and great work. I didn't realize that this scaffold can do so wonders. Was just inquisitive about your slide number 21, where you had shown that the scaffolds help in migration of the cells, particularly neurons. Yes, this one, the part B, A and B. Can you just suggest that is this possibility that they will be able to be helpful in developmental disorders where radial glial cells sometimes do not do their job well for migrating of neurons? Can these be helpful there?
Yes, they should be. The question then comes is, so these are targeted for spinal cord injury where, you would have astrocyte go and then they, don't let the nerves grow in the direction. That's how would you implant these in the application where you are targeting is a big question.
Yeah.
I have not seen many applications. I've seen a lot of application in nervous system growth and nervous system injury for spinal cord, but not for neurological disorder. But if there was an easy way to graft these, they definitely would.
They have the potential to do that.
Yes.
Perhaps, but even if they help in the spinal cord injury, which is a big cause of morbidity in such injuries, I think it will be very, very useful.
Yeah. My ultimate aim is if we can have a system which, surgeons can have in the OR and, because the problem with most systems is like if you use electrospinning, it takes ages to make these nanofibers. If you want to get a scaffold as thick as this, you're looking at, a few hours. Whereas for us, it takes a couple of minutes to make. This is probably sprayed for 2 minutes. So this gives us an advantage. If we have protein-loaded particles, and your polymer solution, you could mix those under sterile condition in the OR and have this sprayed out right away within a few minutes. Whereas if you pre-make the scaffold, you risk that, these polymers are, they are a dynamic system. We see these as solid, but at molecular level, these are polymers. They are always shifting their chains, they're reorganizing. There's always a chance that the protein that is loaded will degrade out over time.
And
So the advantage is if we could, just mix the two components together in the OR and make it in the OR. That's my ultimate aim with these.
Last bit of question, are there any toxicity one should worry about of these particles, or do they self-dissolve later on, or they remain there forever?
So these particles degrade out. They degrade out as silicic acid. That's the main reason we work with these particles, because we have tested in a range of different animals as well. We've just done a study on DSS colitis and asthma model just last 2 weeks. We'll be getting some data on it. We don't see any acute toxicity in any, and we know that they degrade out over time. Depending also, if I make 200 nanometer particles, you put them in buffer solution at 1 mg per mil solution, they are gone within 48 hours. They completely dissolved in silicic acid.
Okay, thank you.
Thank you very much. Dr. Mohit?
Yeah, thank you so much, Dr. Tushar, for sharing your wonderful research. So in your presentation, you have mentioned somewhere this bioimaging thing by using your formulations of biomaterials. So I'm just wondering, so is your platform is just a general platform, it's a specialized platform? So that means you can modulate your platform by tagging some specific type of antibodies or something like this?
Yeah, we can tag. So in this case itself, we've tagged our particle with an antibody that specifically takes the particles to tumor. Here I have not shown the data, but we've also loaded another dye onto it, so we can do both fluorescence imaging as well as the photoacoustic imaging. So we can have dual modalities added to the system. It's a system which is modular. You can add different modalities to it, different functionalities to it.
Tushar, I think he has something from behind. Yeah.
I'm sorry, it was somewhere there. So my second question is about, can we tag something like for example, if I'm kind of interested in immune cell trafficking, for example, I'm just studying movement of the cells from the system to the brain. If we can tag these cells, for example, like mast cells or B cells or T cells, or they are just migrating to the brain at some kind of pathological conditions, because we are getting some kind of pathology inside the brain, but we don't know what are the major players. We are assuming that these are the immune cells which are trafficking from the system to the brain.
Yeah, we can target them. If you have a targeting moiety, we can attach it onto the particles to look at if specific type of cells uptake those or not. I mentioned we've just recently done a colitis study, and in colitis study, the key was that we wanted to suppress specific type of cells, the cells which secrete the inflammatory cytokines.
Mm-hmm.
So that we've done that. We have the cell count number, neutrophils are down, eosinophils are down. So we've looked at different type of cells that can be targeted specifically, but for different application. But if you have an antibody that can be attached, or a peptide, or an aptamer, it's definitely possible.
Okay. So my second question is about that intranasal delivery of this, like any particle or something like this. Since we know that most of the particle or drugs get entrapped in the mucous system when we treat them with the intranasal. So is there any way we can enhance the availability of these drugs or particles to the brain by modulating the surface or something like this?
So the composition of the hydrogels that we have is designed in such a way that there's a mucoadhesive component to it. So the particles, these are not just particles, there's a hydrogel in which the particles are entrapped. And the hydrogels are designed in such a way, there's PVA and chitosan in the hydrogel. I haven't shown the full formulation. And that allows the formulation to stick to the mucus for longer time.
Okay.
And we are hoping that if it sticks for longer time and drug is more soluble, more of it will get uptake. That's what we believe is the case. In the mucosal explant study that we did, there's no mucus layer on top. This is just simply plain washed tissue, human tissue. But I believe we'll see similar type of data, but overall amount could be lower than what we see here.
Okay, thank you so much.
Yeah, thanks, Mohit.
Thank you very much. Any other participant? Okay, so thank you, Dr. Tushar, for giving us your time, valuable time, and sharing your research experience with us. So on behalf of ISF College of Pharmacy and IBRO-APRC, I heartedly thank you once again.
Thank you, Arti.
Now, we have with us our next speaker of the day, and it's my privilege to introduce Professor Pankaj Seth. Professor Seth, his research endeavors after returning to India from National Institute of Health, Bethesda, USA, has been originality of thinking and taking up challenges of direct relevance to the national health problem, particularly HIV-1, drug abuse, Zika virus-mediated neuropathogenesis, and very recently, effect of SARS-CoV-2 on human brain. He was the first to demonstrate that severity of HIV-1 dementia is associated with presence of dicystin motif in HIV-1 TAT protein of HIV-1 subtype B. Further, Dr. Seth demonstrated Zika virus induces microcephaly in infants by altering microRNA circuitry and pinpointed role of two microRNAs. Recently, he started research to understand why COVID-19 patients exhibit neurological symptoms as a brain fog. He has also expressed this possibility in early stage of the global pandemic in April 2020 itself in a comprehensive review published by him, stating that SARS-CoV-2 is more than a respiratory virus that is now accepted worldwide. With these capabilities and expertise, Dr. Seth's laboratory is geared up to understand the cellular and molecular mechanisms as to how the SARS-CoV-2 affects the functioning of brain of COVID-19 patients. His research has been highlighted twice by Nature's India and the International Society of Neurovirology, based in USA, electronic and printed media, reflecting the impact in the field. Dr. Seth, because of his passion to understand national problems which are neglected by others, has earned a distinct reputation among his peers globally. Despite the fact that India has millions of HIV/AIDS patients, less than half a dozen researchers are engaged in studying molecular mechanisms of neurocognitive and motor deficits in such patients, as it is a challenging approach. So Dr. Seth's overcome these challenges with diligence, vision, and scientific approach. He's among a very few researchers who are working in the area of neuro-AIDS in India and also worldwide. He was invited as a collaborator by researchers from Johns Hopkins University, National Institute of Health, University of Nebraska, and other prestigious institutes globally and within India. His research at NBRC has been extensively funded by NIH, R01, R21 Office of AIDS, NIH, US-India bilateral collaborative research partnership on prevention of HIV/AIDS and comorbidities by DSTs, DBTs, and ICMRs. And for understanding of disease pathogenesis, he developed and perfected a human neural stem cell platform of primary cells of brain. The National Academy of Science, India, National Academy of Medical Sciences, and Indian Institute and Indian Academy of Neurosciences have elected him as their fellow. He has been appointed as an area of expert to the number of organizations like DBT, ICMR task force to lend his expertise. He also serves on editorial board of numerous international journals in various capacities. Dr. Seth has served on the board and council of several international scientific bodies, reflecting his research accomplishment, international visibility in the field. He is currently serving as a member of International Society of Virology, USA, International Council member of Society of Neuroimmune Pharmacology, USA, and as a council member of Asia Pacific Society of Neurochemistry. He is promoting the cause of neuroscience in developing countries within India. He made significant contributions for promoting neurosciences with his active role as a General Secretary of Indian Academy of Neurosciences. So we heartily welcome you, sir.
Thank you. Thank you so much for the kind introduction. I was hoping that you would choose something from those rather than listing out the um, entire page. Thank you so much again for the kind introduction. Can you confirm that you can see my slide and hear me?
Yes, sir. It is visible, sir.
Thank you. Thank you so much. So in next 40 minutes or so, what I will try to do is give you a flavor of what we have established here, as was mentioned during the introduction, that we have formed a neural stem cell platform, which we use as a model system to understand how viruses can affect human brain cells and affect the function of these brain cells and cause neurological disorders after the virus infection. We are situated here in the campus, which is a beautiful campus, a green campus, as you can see, in the foothills of Aravalli Hills, around 40 kilometers away from the New Delhi airport. So, Tushar, if you're flying back from Delhi, do drop by at some point, when you visit India. Please stop by and let us know. We'll invite you for the talk at NBRC.
Most definitely. Thank you, Pankaj.
So considering that my audience is mostly students, I have tried to stay away from the data and give you a flavor of what we do at the National Brain Research Center in my lab. So the current research questions that we have here are we're trying to understand how viruses such as HIV-1, Zika virus, and now recently since the pandemic, also to understand how SARS-CoV-2 viruses cause damage to the brain, and the three diseases which they cause, or the disorders which they cause in humans. For HIV, it is HIV-associated neurocognitive disorder, which is also called as HAND, which is the abbreviation for this disorder. And I'm sure most of you might have heard about during the Brazilian Olympics in 2015-16, we had this outbreak of Zika virus, and also increase in the incidence of microcephaly, which is nothing but smaller head size of the infants when they're born. And this happens because when the mothers are infected in the first trimester of pregnancy, which is first 3 months of their pregnancy, there's highly likely chances that up to 7 to 10% of the infants which are born to such mothers can have abnormally small size of heads, which causes or is termed as microcephaly. Now, microcephaly can be with various other reasons, but it has been linked with Zika virus and a strong link. And we have tried to understand how this happens. As I said, we are also interested to understand why people who have were hospitalized during this pandemic and have survived the SARS-CoV-2 infection, especially the long COVID patients, they exhibit problems of their neurocognitive deficits and memory recall and confusion, which is all termed together under brain fog. So I'm not going to talk about this one because recently in May...
Sorry for the interruption, sir. Can you go to the slideshow mode? Because slide is not moving. Yeah.
This is the second slide I'm on right now.
But it is not moving. Only first slide it is displayed.
Okay, let me do that again. Just give me a second. Sorry for it. Okay, just let me know. Okay.
Yes, sir.
Can you see it now?
Not.
No? Wait.
Not yet.
That's strange. Now?
Yes, sir. Now it is
Moving?
We are able to see the title slide, sir.
Only title slide?
Yeah.
Exactly.
Yeah, now it's the second slide.
Okay, let me do it in this mode then. And just confirm now. Can you see the full screen? Second slide?
So not yet, sir.
Not yet,
See
sir.
this?
Yes. Yeah.
Okay, let me try to do it this way.
Yeah,
So,
we can. Yes.
let me know if this moves to third slide, right?
Yes, sir, it is moving. Yeah.
Okay. So what we have done is, I'm not going to talk about the brain fog thing because in May this year we had another school where I had talked specifically about a research on brain fog. I will concentrate on these two research which we have done with these two viruses. Again, how we established the model system and what is that model system and its use for understanding virus-induced neurodegeneration. So this model system which we have derived is of primary cell culture model system, which we derive from human fetal brain cells. And it's a culture procedure which basically has around we get fetal tissue of 8 to 16 weeks of gestation, which we use for culturing. And after removing the meninges and all this is published, we do a series of cell passaging, and finally we have some cells which we call as the human fetal brain derived neural stem cells. And if you look at this picture, this is part of the brain which has been pictured or captured on from day 1 to day 14. And what we have done in this is that you will see that, from you move from this part of the brain, this picture to this picture, you will see that the part of the brain is also grown, as well as it is radiating out or sprouting out some cells which are increasing in numbers from all directions of the brain in culture. And they give out a perfect monolayer, which basically is of human neural stem cells. And again, these are non-immortalized cells and they're primary cells in nature, and of course of human origin, which makes them very relevant for our research. And if you look at this picture little from another angle, you'll see that these monolayer of cells provide a very rapidly proliferating cells which are of great interest to us. Now, we obviously use a specific marker to make sure that they are neural stem cells. For this, we use nestin as a marker, which is a neural stem cell or neuroprogenitor cell marker. And we make sure that they're unspecialized at that stage, at least when they're called as progenitor cells, which means they're negative for markers of astrocytes, which is glial-fibrillary acidic protein or GFAP or TUJ1. Now, the other property that these cells have is they are capable of forming neurospheres, and they form these beautiful sphere-like structures from these neurostem cells in a non-adherent culture. And later on, as their stem cells, they're supposed to give specialized cells, which are basically astrocytes and neurons, or glial cells and neurons from them, if we put them under differentiation. So these cells which we make are actually capable of doing all these things. And that is why we call them as neurostem cells. Now, if you look at this progenitor cells, which is marked for nestin or labeled for nestin in immunocytochemistry, you'll see that most of the cells which are there in this frame are stained for nestin, which was marked with Texas red. And when we differentiate them with providing the neuronal media, you can differentiate them into either astrocytes, which are shown over here by cell-specific marker glial-fibrillary acidic protein, or by the TUJ1, which is a neuronal marker. So we can basically have them differentiate both into astrocytes and into neurons by providing them media conditions and different growth factors. And we've been able to do that, and we routinely do that in our lab. Now, if you look at the structural changes, which happens over the period of time from progenitor cells, when we change the media from neural stem cell media to astrocytic media, which is basically having serum in it, we will see that it changes immediately the structure in 6 hours, 24 hours, and at the end of terminal differentiation, which is a 21-day process, it ends up in giving these beautiful GFAP stained fibrous cells, which are basically indicating that this culture is pretty much 99% positive for the GFAP marker, which is the glial fibrillary acidic protein, the marker for astrocyte. Similarly, we can differentiate them into neurons, and the functions of these astrocytes, as we know, are immense, and I'm going to go into those details, and that is why we're trying to differentiate both astrocytes and neurons from them for their use later on in our study. So if you look at this, again, a beautiful structure in culture. This is how astrocytes look in our hands, the star-like morphology, the fibrous structures, and so on. So again, because the animations are not working, here you can see that the progenitor cell basically has a different structure, and at the end of it, when we have, after 21 days of differentiation process, we have this TUJ1 or beta-3 tubulin positive neurons in our hands. And these are pure populations of astrocytes and neurons, and then later on we can also bring them together to make the co-cultures, which we have also done in our hands. Now, the neurons which we make, we have also checked them for their types, and we have found that they are Th positive cells, which is basically staining for tyrosine hydroxylase, which shows us they have dopaminergic, they're kind of dopaminergic neurons. And if you look at this merged image over here, this shows you that most of the cells which we have are actually Th positive, and these are 3 different preparations which we stain at different times. And if you look at here again, they are Th positive TUJ1 neurons, which means they can be also used and are relevant for the Parkinson's disease research. In fact, we have collaborated with people over here at NBRC, and these cells have been used for understanding Parkinson's disease as well. Now, again, the advantage of these cells is that they are primary cells, so they are non-immortalized, and also since they are of human origin, whatever we do in animal studies, we can actually validate those things in these human neurons. And that's what we have been doing. Now, as I said, this because these neurostem cells have the potential to be differentiated into neurogenesis or into neurons. What you're looking here is a differentiating neurosphere, which is basically a collection of cells. And if you stain them while they're undergoing neurogenesis, you can see that they are positive for neurons. This is DAPI. This shows the number of cells which are present over here. And you then see a population of cells which is basically green, red, orange, which means that some of the cells have completely differentiated while the others are undergoing the process of differentiation. And they co-localize both for the neuronal marker and nestin marker, which means that at the end of the differentiation, the most of the cells will be green in color, indicating again that they are differentiated into neurons. Are my slides moving?
Yes, sir, it is moving.
Moving, sir.
Then we had a very energetic and a very ambitious summer student which came to the Indian Academy of Sciences to us, and he wanted to check if the cells have tri-potential. In other words, whether the neurostem cells which we made can actually also be differentiated into oligodendrocytes. And what he did was, again, we changed the conditions of differentiation, and we added different growth factors. We gave media which would enhance the differentiation of these progenitor cells into oligodendrocytes. And I don't know if you can see at your end, but we had some structures which were showing oligodendrocyte-like morphology, which was later also stained and confirmed by immunocytochemistry. And we had these oligodendrocytes which were positive for O4, which is a marker for these oligodendrocytes. And we also did NBP and other markers, and we confirmed that these cells actually have the potential to be also differentiated into oligodendrocytes in addition to their capacities to become neurons and astrocytes. So the differentiation of the end-stage cell depends on what media conditions and growth factors we provide to these neural stem cells, and they typically take around 21 days to differentiate into the cell type that we want. So using this model system, we have basically tried to grow them both as monolayer cells, which is shown over here, or as suspension culture as neurospheres. And the both different these cell systems of same cells of neural stem cells have advantage in different research questions what we ask. And using these cells, we have basically published several papers which shows that this model system is very well accepted in the field, as well as has been very useful in understanding various neurological disorders. So most of these papers are from our lab as well as from collaborations, and 2 of our papers have actually featured in the Nature India, which indicates that the research which we have done through the cells have actually been of great value. Now, if you can look at this, how the neurospheres are formed. The one single cell basically divides into 2, 4, and 16, and then after 48 to 72 hours, we have this sphere-like structure. So, can you see the sphere-like structure or the animation?
Yeah, we can see the sphere-like structure, sir.
Yeah. So the beauty of this sphere-like structure is, so at the end of this 72 hours or a time point, what we decide, we can actually, we know how many cells we plated in a 96 cell plate or a 24 cell format. And then if we want to see whether the cells are proliferating at the same rate or in the presence or absence of a test agent, which can be a viral protein or a cell proliferation kind of a compound, we can at the end of the stage decide by measuring the diameter of this sphere. We can see if we have a bigger sphere, which means that agent is helping in cell proliferation. On the other hand, if we have a viral protein, it is decreasing the diameter of these neurospheres without, in the presence of the viral protein, it shows that it is anti-proliferative. So using this model system, we have also understand how the test agent is good or bad for the proliferation of the neural stem cells. One can measure the diameter as well as disrupt these neurospheres, bring them into single cell and count them, and again also find out at the end of 72 hours, compared to control, how many cells they proliferate into. So by using this model system, we have been able to understand if test agents are good or bad for the neural stem cells. Now, again, since they have potential to differentiate, this is another slide which shows the neural stem cells undergoing neurogenesis or formation of new neurons, and the new neurons are indicated by the presence of this green stain, which is marker for the neuron marker TUJ1, and we did also beta-3 tubulin to confirm whether these cells are actually neurons which are formed. And again, the yellow and orange cells are basically cells which are undergoing differentiation, have not completed, since they are co-localizing both for the nestin, which is marked in red, and the green marker. So the co-localization gives you the yellow and orange color. And again, at the end of 21 days, and sometimes a little later, they give rise to all neurons in the culture. Then we had we became bold in our approaches, and we started, once we had successfully differentiated human neurons and human astrocytes from these neural stem cells, we started making co-cultures of neurons, because astrocytes and neurons never are present in the brain in isolation. If you want to understand the real function of these cells, you have to bring them together, because neurons cannot function at all without the presence of astrocytes. Or in other words, neurons depend on astrocytes for a lot of things, including growth factors, as well as the shuttle of neurotransmitter which is being taken up from the synaptic sites. So we have started now doing co-cultures, and how we make these co-cultures, we take astrocytes and neurons which are differentiated separately. We plate them into 2:1 ratio. We take 2 of astrocytes and 1 of neurons, because astrocytes are always, they outnumber the number of neurons. So we try to simulate the conditions in the brain, and we have always more astrocytes than neurons. We plate astrocytes and then put neurons on the top, and then allow them to give them 6 to 7 days to form these junctions and crosstalks between them, and then we use them for our study. So one such example of the co-culture is seen over here, where you see these red cells, which are again marked for the GFAP, the astrocytes, which is glial fibrillary acidic protein, and you see a green neuron which is extending its processes to the neighboring astrocytes. And we have again more astrocytes than number of neurons. And once we have this co-culture structure in place, then we try to use them for our study. An example of that is shown over here. When you add HIV viral protein TAT into the culture system, you will see that this expanded over here. Here's a neuron, here's an astrocyte, And this neuron, which was forming a connection over here to astrocytes, actually retracts and provides this bead-like structure, which is shown by the yellow arrow over here, which indicates that this neuron is about to die. And if we do not wash off TAP protein from this co-culture system, these neurons will actually end up going under apoptosis. So using this model system, the potential of this model system is immense, and the model system, again, and I'm talking about, is of neurostem cell of human origin, primary cells. We have used this to study how HIV affects the stemness of neurostem cells, and I will give you a little detail in summary slides in subsequent slides. And we've also studied the effect of HIV in neurogenesis, and effect of HIV in astrocytes, effect of HIV on glia-mediated neuronal degeneration, on Zika viral proteins of neurostem cells, and we've extended this model system also to understand how SARS-CoV-2 proteins or the virus is actually causing neuropathogenesis, which is resulting in the brain fog in the long COVID patients. So very quickly, we all know that there's a very robust blood-brain barrier, and what you're looking at is a cross-section of blood microvasculature, and you see RBCs inside. This is the brain part, this is the blood part over here, and if you pay attention over here, you'll see that there's a lining of this capillary where you have these endothelial cells, and then you have these astrocytes which provide the end feed, which basically kind of encapsulate the blood microvasculature. And this is how they take all the goodies of oxygen and glucose for the neurons, and they provide them there. And also they dump what they do not require in the blood. And so the astrocytes play an important role in the blood-brain barrier pathway. Now, why I'm telling you this, because this has relevance when we understand the neuropathogenesis caused by the viruses, because all these beautiful structures which are provided by the nature are disrupted in the presence of the virus. And what happens, because virus like HIV travel in the blood and they get access into the brain by disrupting this structure of blood-brain barrier. So if you remember this picture, which was shown in stories when we were studying in our junior classes, this is basically a horse where you have soldiers inside, and there were two emperors which were fighting for a battle, and the one emperor had a very strong fort which was very hard to permeate. And then what happened when the other emperor got an idea that he will present a horse to the emperor, showing that I came as an enemy, but you won my heart, and I want to present you a horse or multiple horses. So these were wooden horses where he had hid his soldiers, and the other emperor opened the gates for him, and this horse were accepted. But later he realized that this horse actually had brought in enemy soldiers into their fort, and this then permeated the fort, which is basically breaking up of the blood-brain barrier. Now, let me show you in an animation over here if it works. That you're looking at a cross section of HIV, and then this is basically the enemy, which basically can get into the blood circulation by various ways. Once it gets into the human circulatory system, it also gets access into the blood vasculature. And what you're seeing over here is a cross section of the capillary. You see RBC, you see this blue cell or purple cell, which is a monocyte, a blood cell, and a very small white particle, which is basically the viral particle. And in the cross section, you also see that the lining of the endothelial cells, which do not permit this virus to get into the brain parenchyma from the blood because of the blood-brain barrier. Now you will see later in this animation how these viral particles will breach this blood-brain barrier. Now also point to be noted over here is that this blood-brain barrier is basically a selectively permeable barrier, which means that it selects what is allowed or not allowed from the blood side to get into the brain side. And this selectively permeable barrier basically has given privilege for this cell, which is the monocyte, to access the blood from blood to brain. Because monocytes go as surveillance cells in the brain parenchyma, because the brain does not have an immune system of its own, So it provides access to these monocytes for the good of the brain, so that they can come and look, and if everything is okay, and if there's some problem, they will take care of it. Now, what the virus has learned to do is they have the capacity to go and hide in these monocytes. Similarly to the previous slide where I showed you, so this is the wooden horse, these are the army, the enemy soldiers, which will go in this horse. And because this horse has the privilege to go through the blood-brain barrier, this is how the viruses enter into the brain. So you're looking at here the monocyte which is infected with the HIV particles, while the small virus cannot go. A bigger cell as monocyte can pass through the blood-brain barrier. It brings these small viral particles inside, and then it disrupts, and the spikes, which are the GP120 protein of the virus, it binds to the CD4 receptor, and what you saw is an infection of a brain cell inside the brain. And these cells which have the CD4 receptor as well as the chemokine co-receptors, if they have both of these cells, the virus can infect HIV. Similarly, what we have seen in this recent pandemic by various animations, that how SARS-CoV-2 uses the ACE2 receptor to infect cells. So similarly, HIV uses CD4 receptor and it gets into the brain cell. Now, once it gets into the brain cell, what it does is basically it causes HIV causes dementia by affecting the functional unit of the brain, which is basically both neurons as well as the astrocytes. Now, memory recall problems occur in the AIDS patients, and the failure to do executive functions and poor quality of life is what is called as HIV-associated dementia. Now, how it causes is shown over here. So what you saw in the animation was entry of the viral particles into the brain cells, and once it comes into the brain cells, there are two ways by which it can infect the neuron. It can damage the neuron. Now, interestingly, neurons do not get infected by the HIV viral particles, but they do get affected, which means they undergo apoptosis process, and they do so because of R in the viral structure. They are shed off, the both GP120 and the TAT protein, which I showed you also in the co-culture. They are neurotoxic in nature, and they're secreted with these viral particles when they are in the brain. And these viral particles can directly cause apoptosis of these neurons. So again, without causing infection, they can cause damage to the neurons because the viral proteins are secretory in nature, and they are present in the astrocytes, as well as the viral particles are present. Viruses are present in the microglial cells where they multiply, and then they give out these proteins which are directly toxic to the neurons. The other way is indirect pathway of death of the neurons, which is caused by the effect on the astrocytes. As I told you that neurons depend on astrocytes for a variety of things such as growth factors and the glutamate uptake and clearance of the glutamate from the synaptic site. When astrocytes are infected with HIV, they stop doing their function at the optimal level, and which causes the not providing of growth factors in optimal levels, and the clearance of glutamate from the synaptic site is also affected, and that is when it causes excitotoxicity at the synapse, and it also again an indirect pathway by which astrocytes mediate the neuronal damage because of the virus, because astrocytes stop providing the support to the neurons. And so this is the way, direct pathway or the indirect pathway by which HIV causes damage to the neurons. Now, from using this model system as astrocytes, neurons, co-cultures, and neurostem cells, our lab has contributed significantly in understanding of how HIV causes dementia in individuals, AIDS patients. What the initial question was whether there's a low incidence of HIV-associated dementia in India. We had found that with the TAT protein, which I was talking to you, which is toxic, the strain which is found in India is of subtype C, and we found that there's a mutation in this TAT protein which causes lesser toxicity in Indian patients. So the level of dementia, if you compare to the Western population, in Indian patients, despite the fact that there's prevalent and a lot of cases of HIV-associated deaths in India and AIDS cases in India, the consequences on the neurological aspect in India is lesser, and it is because of this toxicity of the TAT protein. Later on, we asked another question using the neurosphere assay whether the TAT protein is good or bad for the neurogenesis and the proliferation of cells. And what we found was in the absence of the TAT protein, which is a toxic protein, if you do not have it in culture, the neural stem cells, as they should, they divide very well. They have this Ki67 positive nuclei, which is indicating they are proliferating, and they have this cyclin D1, which is again a cell cycle regulator, stained very well. If in a parallel plate, which was coming from the same parent neural stem cells, if we add TAT protein in 24 hours, you will see that the number of pink nuclei, which is indicating presence of red Ki67 cell in the DAPI positive cell, the numbers are lower. The green stain is almost gone, which is indicating that cyclin D1 level is down, which then results in stalling of the proliferation or moving of these cells into the next phase of cell cycle, and which is indicating there's a decrease in the proliferative rate of these neural stem cells if HIV TAT is present. So what is that indicating? This is indicating that in the presence of TAT protein or the virus in the brain, the neural stem cell proliferation is decreased. Now, Meher took this work further, and she basically gave more insights into how the proliferation of these neural stem cells is affected by the HIV. And she showed that the TRIM32, which is a stem cell fate determinant, and the microRNA 155 play an important role in TAT-mediated decrease in the neural stem cell differentiation. This paper was published in 2016 in Cell Death and Differentiation. So using again this model system, I'm trying to tell you how this model system, once it was established and confirmed that it is neural stem cells, we've used it in understanding how different viruses, till now HIV is being shown, how they affect brain cell functions. Then Meher later also showed very elegantly that how neurons are damaged because the damage to the astrocytes. So what happens when she used the co-culture system, and what she showed was in the presence of the TAT protein, what happens, there's an enhanced increase of ATP generation. And this ATP source was found to be coming from the glial cells. And she showed that if you block the voltage-dependent anion channels of the astrocytes, you can actually prevent the death of the damage by apoptosis of the neuron cells because of TAT. So in other words, and also we had found that microRNA 320A plays a role in this activation of the VDAC. So if the TAT basically blocks the VDAC, microRNA 320A, which then blocks the VDAC, and then because of that you can prevent the enhanced cell death, which is caused of the neurons because of excessive increase of the ATP release from the astrocytes. So again, another example how this model system was helpful in understanding HIV-induced neuronal death. Manju, another student in the lab, also showed using the same model system that indeed it is the P2X7 receptor, which is the prenergic receptor, which helps in TAT-mediated release of these different molecules which are damaging to the neurons from the astrocytes. So the astrocytes, which is supposed to be friend and supporting the neurons, actually turn out to be enemy of the same neurons because they are agitated, because of the infection, because of the viral protein, and the various mechanisms by which they cause damage to the neurons have been worked out in the laboratory, and this has helped in designing different molecules by controlling or plugging these different receptors or channels by which we can prevent neuronal death in the AIDS cases. Further taking this work on, Chitra, another student, has shown that how HIV TAT-B is increasing the efferents which are important for maintaining the glutamate regulation at the synaptic site. So actually, as I told you earlier, the presynaptic neuron actually gives out the glutamate neurotransmitters, which are taken by the postsynaptic neuron. This is in the normal brain condition shown in blue over here. And all the excess of the glutamate, which is not utilized by the postsynaptic neuron, is taken up for recycling by the astrocytes. Now, this function of astrocyte is affected in the presence of TAT protein on AIDS cases. What happens because this uptake of the excess of glutamate particles, which is shown in red over here, is stopped or decreased, due to which there's build-up of this glutamate, which actually is then harmful, and that causes damage to the synaptic site, and both the presynaptic and the postsynaptic neurons are damaged because of glutamate excitotoxicity, which is caused because of decreased uptake of this. And efferents have a role in this. This has also been proven using the same co-culture system which we developed in our laboratory. And last slide in HIV is shown over here, where again another mechanism by which we have shown how astrocyte-mediated neuronal damage can be prevented, because we have worked out the entire mechanisms by which the TAP protein is causing reactive astroglia, which is leading to neurotoxicity. Now switching very quickly to the Zika virus, and just few more slides, I will show you how Zika virus causes infection. Zika virus infection in the pregnancy causes microcephaly. I'm sure most of you have also seen this slide in the popular media, where it shows the normal head size, and this is why the mothers are supposed to go for their regular checkup during the pregnancy, and every trimester they measure the circumference in the using ultrasound, they measure the head circumference of the developing fetus in the uterus. And this is the normal size of fetus, but in the cases of Zika virus, you can have severe microcephaly, and you can see how the head does not grow, and you see abnormally smaller head size. Now, why is this happening? Now, during development, if you understand development, you will know that during development, the neural stem cells are proliferating at a very high rate, and they are not only proliferating, they are also differentiating into astrocytes, neurons, and forming the structure of the brain at the head. Now, this process, if this is disrupted, this will obviously lead to the cessation of the proliferation or differentiation of these cells during development during the first 9 months, and may lead to microcephaly. Now, with this background, we started in our lab, and whether this is actually happening or is it real is shown in this picture. As you see, the kids which are born with these abnormalities, small head size, most of them die at the early stage of their life, but those who continue to grow and are saved, they have very bad future because their brain is very, very underdeveloped, and they have actually mentally retarded. So this problem is very bad, and it does happen, microcephaly. So what are the reasons for that? So to understand this, we looked at the viral protein, and since there was association of microcephaly and Zika virus outbreaks in the regions, we looked at the viral proteins, and what we find is that this virus has the structural proteins as well as non-structural proteins. So we took some structural proteins and some non-structural proteins and put them in our neural stem cells, and we found that the envelope protein of this virus basically prevents the proliferation of the neural stem cells. And we found that it also, in addition to preventing the proliferation of these neural stem cells, which we studied using that neurosphere assay, which I showed you earlier in the animation. And we also looked at the migration of these differentiating neurons from these neural stem cells in the presence or absence of the E protein, the envelope protein of the virus. What we found, and you can very elegantly see, in the absence of the E protein in normal situation, you will see that the neurons are sprouting out and are dividing and are migrating from the place from where they are coming out. But in the presence of the E protein, if you transfect this E protein into these neural stem cells, you will see that this migration as well as differentiation is faulty in the presence of the E protein of the Zika virus, which is also quantitated over here, and you see a significant decrease in the migration of these new neuronal cells, which shows you that the proliferation and differentiation is affected, which was published by Reshma very elegantly, again in a very high impact journal, which is Cell Death and Differentiation, where she showed that Zika virus E proteins actually alters the microRNA circuitry, which basically is important for the development or neurogenesis during early stages of development. And she showed that 2 microRNAs, which is 2043P and 127, which actually have their targets on Notch 2 and PAX3, which are important for stemness. These are affected, and due to these, you find that the migration and proliferation of the neural stem cells is affected in the presence of the Zika virus E protein. So this is what we have published in the Zika virus features. Now, during this period of years, I'm coming towards the end of my presentation. We have developed this neurostem cell platform, as was mentioned during the introduction. And the beauty of this model system is, again, is that it is grown as monolayer, as neurosphere, as well as co-culture. And we have now also branched out and also established induced pluripotent stem cells from human blood, where we also differentiate neurons and glial cells from these iPSC cells. And we have entered into various collaborations, both international and national, because of this model system, which has helped in understanding a lot of neurological disorders, including by the viruses, as well as in Parkinson's disease and Alzheimer's disease. And so the conclusion is that we have several lines of these human fetal brain-derived neurostem cells which have been made by NBRC. It's a unique resource for the neuroscience community in India. We share these cells with various organizations and neuroscientists in India, and they have also been able to answer various questions, research questions, using these primary cells of human origin. We have well-standardized protocols of differentiating astrocytes, neurons, and oligodendrocytes from the neurostem cells. And this model system has helped to understand effect of various test agents on neurostem cells during differentiation, neurogenesis, gliogenesis, and in fully differentiated cells as well. With that, I am very thankful for the patient audience. But I'm sorry for the technical glitch which happened, which I don't know why. I've given several of these talks before on Google as well as Zoom, but this was the first time I had this problem of presentation. So I'll be happy to answer any questions if there are any.
Thank you very much, sir, for wonderful talk. Now the session is open for discussion.
Yes, Mohit?
Yes, Dr. Tushar.
No, let Mohit ask. I think he raised it.
Okay. Thank you so much, sir, for sharing this wonderful research. So my question is about, since I am the microglia guy, that I'm working on the microglia a lot. So you have tried to differentiate these cells on astrocytes, on oligodendrocytes, on neurons. So have you tried ever like converting these neural stem cells toward microglia?
So the microglia cells are not brain resident cells. Their origin is entirely different. So these neural stem cells will not make microglia at all. This is for sure. So there's no point trying it. Microglia cells, the best source of that which people are now using are the iPSC cells. So and those are commercially available. If you can get iPSC cells, and they're very well-defined recipes and media conditions, from iPSCs you can derive microglia cells. These neural stem cells will not give you origin to microglia because their lineage is entirely different.
I'm sorry, I lost my connection.
So did you get my answer? So I said,
no.
So to get microglia, the best source would be iPSC cells. And iPSC cells are commercially available, and also differentiation protocol for microglia are also published very well. So people are now relying on iPSC cells for getting, if you want human microglia, you can take iPSC cells, differentiate them into microglia. Neural stem cells, because the origin of microglia is mesodermal and these are a different origin, neural stem cells will not differentiate into microglia.
Okay.
Tushar?
Thank you, sir.
Thank you, Pankaj. It was amazing talk. A lot of data, a lot of fantastic data. My question was around you're differentiating neuronal sphere into specific type of cells. I assume that you're using growth factors, anabolic growth factors. Have you tried any, surfaces which, one of my interest is to look at, micropattern surfaces. Can they themselves guide, phenotypical differentiation of specific type of cells? Is that something that you've tried, or are you,
I'm interested. I haven't tried, and I'm glad my talk and your talks were today, and I could attend your talk, which basically did. I have read that these surfaces can help in targeted differentiation. They do help, but I didn't find a collaborator, and I was a bit more engrossed in what we were doing in the neuropathogenesis of these viruses. I could never get a chance to think about and grow on that area, but we can, definitely we should touch base, and this is why these meetings are very important. You get to know people, and you see the slides. If I have read your paper, Tushar, I might not have thought about collaboration, but now that I've heard your talk, maybe we should build this up.
Yeah, because that's something that we are already looking at surfaces to guide macrophage, polarization just based on surfaces, and we've done that. Like just by changing the surface features, we can guide, but we haven't tried, whole cellular phenotypical differentiation, and that's something that I'm really interested in. And that would be, we have capacity to do patterning various types, but yeah, we can chat after this.
Sure. By email or something, and then I'll find your email and you can find, and then we'll write to each other. Are you visiting India anytime soon?
I was planning to, but not at the moment. Perhaps
anytime when you come, we can visit NBRC. We, as I said, if you're landing in Delhi, we will just pick you up from there and stay here for a day or two, and then you go wherever you want.
That's fantastic. Thank you so much. The other question I had was, how long does it take, to get the cell differentiation to happen? I'm assuming it's quite a long process.
21 days. 21 days. Yeah.
And it's for both astrocytes and the neuronal cell.
True. So, they start giving these markers of, let's say, if you're talking about astrocytic differentiation, the GFAP starts appearing from day 6 or day 7. But to get all of them differentiated into them, and then having other markers and their functionality, which we study by release of chemokines and cytokines from these astrocytes, it actually comes best when they complete after 2 weeks. And we typically have decided 3 weeks. And it's not difficult to culture them for 3 weeks because, you just put them and you keep changing media. You do have to split them in between once or twice, particularly astrocytes. Neurons do not require splitting as they grow because they don't divide. So the initial differentiation happens in neurons, and then after the second split, they don't require a split. And that is why when we have to do any neuronal work, we plate them in the second split in this culture dish where we want to do our experiments, because we know once we replate them, they will not attach at all. It takes typically 21 days. So we have decided 21 days to keep that. And then, every batch when we differentiate, we again check for the cell-specific markers. And now we do it pretty routinely. So it's almost automated.
Thank you. Can I ask one more question, if you don't?
Charan has to allow that. I am okay.
If that's okay,
yes, please. Yes, please.
So the 2 microRNAs you mentioned. So how is so right now microencephalus or forgetting the term. Right now, are there any so is the ultrasound the only way to detect it? Is there any other way to detect this disease even at an earlier stage of fetal development?
So I think the routine way by which usually this is done across, even in India and even in primary healthcare centers now, ultrasounds are available. And the mothers anyway go through this routine checkup. And the other beauty is that if the infection has occurred in the first trimester, only then it is a threat of microcephaly. If this happens after second or at the third trimester, at the end stage of the pregnancy, then the chances of microcephaly are very low, because the entire development of the head has actually happened by the third month. So one has to worry if there's a monsoon season and a dengue and Zika virus outbreaks, and if a mother is showing any symptoms, then definitely one has to look at it very carefully. Now, fortunately enough, Zika virus is not a threat anymore, because most of us are seropositive and we have developed, kind of herd immunity against it. But there are, somewhere papers still popping up where they have done long-term studies on the kids which were actually born normal with head size, but they have neurocognitive deficits. So they do kind of lag behind in their developmental milestones, and they're not as sharp or as good as their other classmates are. And these studies have now raised more, what you say, roll eyes because of is troublesome because if you find the child born with a normal head size, that doesn't mean that he's okay, because you will know about this a little later. So if one has to go into neuroimaging and one has to do the DTIs and look at the tracks, if the hardwiring is problem, then you may have problem. Now again, the smaller or the subtle differences which may be caused by the virus may not be picked up as real symptoms. But again, that's why detailed studies are now into progress, and some people are trying to see if these kids which are having problem, if they were done, any scans were done on them by chance or something. So they're going to backtrack it. But still we don't have another outbreak, nobody needs to worry about it that much. But it's always good to know why these damages can be occurring, so that if you know this, one can perhaps intervene at these steps.
Perfect. Thank you so much, Pankaj.
Yeah, Dr. Verma.
Thank you very much. Now we have now we have Dr. Neeraj Verma. She want to ask some question.
Sure.
Good morning, sir. My
Yes,
question
Dr.
is,
Neeraj.
is the is the neuronal stem cells is able to treat the multiple sclerosis also, sir?
So since the whole field was working with using neurostem cells as a therapy, we took the other path. We always go away from the crowd, and we started using them as a model system. Now, neurostem cells have been tried and experimentally, and they have a lot of promise for causing or providing therapy for everything on the earth, but they have actually not been able to cure anything. Now, neurostem cells or stem cells in general have been found to be helpful in some diseases because of the trophic factors which they secrete. Multiple sclerosis, can it be cured by neurostem cells? It's all experimental, has worked in some animal models, but I don't see any standard cure or as a procedure for cure which has been used, where stem cells have been used for curing multiple sclerosis. These are all experimental and have not been cleared by FDA or any other big agencies. They hold promise, but they're not.
Okay, thank you, sir.
Thank you very much. Dr. Mohit want to ask something?
Yeah, I have one last
Yes,
question.
Dr. Mohit.
Quick question. Since we are talking about this organ system, so my question is about the human organoid system. So do you think in near future we can replace the in vivo studies by using human organoid systems for the translational
push
research?
to stop animal research, and for both reasons, for ethical reasons and also for reasons that most of the molecules which were showing promise in animal studies fail when they come into human systems. So human organoids are seen as a very potential replacement for animal research or in vivo research, but you should understand that they are again in isolation. The advantage of animals is because then you have the liver, brain, spleen, everything working together. You have a holistic system. Well, if you want to study in isolation, well, human organoids is a good, kind of alternative, because you now have organoid of everything almost. You have intestine, livers, lung, brain, everything. But
at least we can reduce the number.
But there are problems associated with it because at the core of this organoid is only up to a size you can grow it. At the core, they are again hypoxic in nature because and now people have been able to develop blood vessels into them, and they are coming with more advancements. But this is again something what we found when iPSC came. We found iPSCs will cure everything because now you can differentiate anything. iPSC failed. First came embryonic stem cells, then came iPSCs, now brain organoids. They will not There will be good model systems to study, but I don't think one can expect them to cure diseases. And I think for brain organoids, most of us do in the field understand that they are good model systems only, and nobody is using them for curing any disease.
Yeah, at least we can reduce the number of animals that we generally use for our, like, for even for identification of some lead or something like this.
Yeah, so what people have now been using is making humanized animals. You know, if you want to study spleen or you want to study brain, they put these cells and make them human-like structures. So you can still try to study a disease in holistic, but again, they're nowhere close to what actually is happening in disease pathogenesis.
Okay, thank you so much.
Thank you very much. Dr. Alka. Yes, Dr. Alka.
Please,
thank you, sir. It was a very informative talk. Sir, I want to know what is the best protocol for culturing neural stem cell?
Can you write to me, Charan? Can you give her the email ID, or you give?
Sure, that's what I was. I was speaking in between, so I'll get in touch with this particular question.
Okay, sir. Okay,
you can write to me, Dr. Alka. I will send you the protocol.
Yes, sir. Sure. Thank you. Thank you.
So, any other participant?
I think
Dr. Charan, you want to share your views? Yeah.
Already, I think a lot of questions has been taken and discussion is done. So I think I should restrict myself. Otherwise, I had one, but later on, I will be in touch with you.
Charan, you can call me later.
Yeah, sure, sir. Thank you so much.
So thank you very much, sir, for sharing your views and your research work with us. We feel privileged. So thank you very much, sir.
Now with sir, please.
We have with us Dr. G.D.Gupta.
Thank you very much, Dr. Arti and Dr. Charan. So first of all, I am sorry because some of the college engagement, I was working in the another part, so I was not attending the lecture. But after the end of this, I feel that both of the today's expert talk, no doubt, obviously very excellent because huge interaction with the participants. So thank you very much, Dr. Tushar and Dr. Pankaj for interaction and